Okay, everyone, our first class for microbiology, BIOL 2010, is just an introduction to microbes. So we want to sort of lay the groundwork. What I want to do is make sure we all know some basic vocabulary and some basic concepts before we delve deeper. And a lot of what I'm going to talk about here in this chapter, we will be revisiting as we move on. Okay, so you can see here, this is chapter 1 in the book, Introduction to Microbes. Microbes are indeed all over the place. All right, so what we are studying is microbiology this summer. Microbiology is the study of microorganisms, which are living organisms, but those that are too small to be seen without the aid of a microscope. So we call organisms too small to be seen without a microscope microbes, sometimes germs. Way back in the day, people called them animalcules or wee animalcules, cavorting beasties, sometimes bugs, things like that. So they have different names, but today we refer to them as microbes or microorganisms. Now, microbiology also involves the study of cells. Cells are the smallest things that are themselves alive. Cells basically a membrane surrounding liquid and other material on the inside. If something's not a cell, it's not considered living. So, what are the types of organisms that we study in microbiology? Well, there are a number of them. From your previous biology courses, you might remember the term prokaryote. Prokaryotes or prokaryotic cells, which we will be revisiting, are the simplest type of cell. No nucleus, not a lot of internal structure, things like that. Now, in microbiology, prokaryotes include bacteria and archaea are your two big groups of prokaryotes. Eukaryotes are organisms composed of more advanced cells. These are cells that have a nucleus and have a more structure internally. This includes protozoans. Protozoans you might have heard of as plankton. These are little single-celled prokaryotes you might find in pond water or in the ocean. Fungi are molds and yeasts. Fungi also include mushrooms, but mushrooms wouldn't really be considered a microorganisms, but molds and yeasts are. Algae is a eukaryotic organism that many of them are microscopic. And parasitic worms. We won't touch on them too much in here, but parasitic worms are considered part of microbiology. We also have organisms that are acellular. Now, prefix A, not cellular. So these are things that are called viruses and prions. So viruses and prions are infectious proteins, are not considered living because they're not a cell. All right? So here are some examples of some things we study in microbiology. Everything from this algae down here to viruses to bacteria and protozoans. Okay? Now, microorganisms are hugely abundant across the surface of the earth. Microorganisms are associated with other living things, especially you. Or one of the things is other living things. So your normal microbiota, sometimes referred to as your normal flora, these are microorganisms that normally live or reside on or within your body. You have many more bacterial and microbial organisms in and on your body than you do human cells because they're so small. And we'll be revisiting this later in the course. But part of your normal flora or normal microbiota is bacteria. That's the big one. Most of them are bacteria. But there's some archaea there. Some protists, fungi, and even viruses can be part of your normal flora. They live on or in your tissues, fluids, or even the surface, like on your skin surfaces. Now, the good news is these normal flora, they do not normally cause disease. So under normal circumstances, they don't cause disease, but some of them could cause disease under certain conditions. And again, we'll revisit that a little bit later in the course. The members of your normal microbiota that can cause disease under certain conditions are called opportunistic pathogens. More good news, your normal microbiota, these organisms are often very beneficial to your immune system and overall good health. Some of you may have heard of, quote unquote, good bacteria or probiotics. That's what we're talking about here. They come in kind of two categories. We have resident microbiota. These are permanent, lifelong members of your normal microbiota. They're there all the time. And then there are transient microbiota, which are non-permanent residents, kind of just passing through. You might pick up transient microbiota as you touch surfaces or eat food or something like that. But they don't really stay around for too long. They might attempt to colonize and become part of your resident microbiota, but they're unable to remain for long periods of time. And we'll talk about why a little bit later in the course. Now, besides your normal microbiota, microorganisms, again, up here at the top, microorganisms are associated with other living things. They can cause disease. Disease-causing organisms are called pathogens. Microbes that don't cause disease then would be a non-pathogen. And the human microbiome refers to all the genes of all the microorganisms that make up your normal flora. So a collection of those genomes, all those genes. All right? So one of the things that microorganisms do is they're very much associated with other living things. They are also involved in many activities. Now, perhaps the biggest one is decomposition, breaking down dead organic matter, decomposition. Fermentation. Talk more about decomposition in just a minute. Fermentation is where sugar is fermented and converted into, various byproducts, or ethyl alcohol being a big one. So fermentation is responsible for a lot of the products pictured here, okay? So we're going to convert sugar into ethyl alcohol or some other types of products. Oops. Infection is another thing, of course, that microorganisms can be involved in. If you are pathogenic, you cause disease, and that's an infection. Microorganisms often produce useful byproducts, including medicines, chemicals, antibiotics, vitamins, things like that. They can be heavily involved in agriculture, like we see pictured here. Bacteria, for example, are responsible for pulling in nitrogen from the atmosphere and then converting it to a form usable by plants. It's a process called nitrification and nitrogen fixation. Very important, again, something we'll get to a little later. And then there are many others that we'll be touching on. Now, again, of particular importance is decomposition. So if we look at that in a little bit more detail, decomposition refers to breaking down dead organic material. So if a living thing dies, all those nutrients that are in its body are still there. They don't just go away because the organism's dead, they're still there. So decomposition is where dead organic material is broken down into inorganic nutrients, and then we also release some carbon dioxide and energy in that process. Decomposition is often referred to as recycling nutrients back into the ecosystem. So when a dead organism decomposes, the nutrients that were in its body are put back into the ground or back into the air or back into the water and recycled, and they can be reused by plants and other forms of life. So once those nutrients get put back into the ground or into the air, again, the nutrients are usable by plants, and then something eats that plant, and there's a transfer of energy and nutrients there, and then something eats that animal, and on up the food chain. So we've recycled nutrients. Decomposition returns carbon dioxide to the atmosphere and releases energy in the form of heat. It also provides energy to the decomposers that are breaking down the dead organic matter. So because it provides energy, it's a form of what we call cellular respiration, a process by which energy is produced. And we'll get to that later in more depth. Decomposition is a complex process with many key steps and many different organisms involved, some of which are pictured here. Now, a very important point that you want to remember is bacteria and fungi. These are the two main groups of organisms that are responsible for decomposition. Okay? So, we might think of decomposition as this. You see this guy here, he says, just think, here we are, the afternoon sun beating down on us, a dead, bloated rhino underfoot, and good friends flying in from all over. I tell you, Frank, this is the best of times. So, we might think of decomposition that way, but it's really bacteria and fungi that are doing the decomposing. All right. Now, if we shift gears a little bit, it's important to keep in mind that microorganisms are too small to be seen without the aid of a microscope. So, a little bit of history here, discovery of cells and microorganisms. Now, the microscope was actually invented back in the 1590s, so it's a lot longer back than people think. This is a replica of the first compound microscope. It could magnify 10 times. Fast forwarding a little bit after that, we have our first key figure from history. This is a British biologist named Robert Hooke. Robert Hooke is the first person to discover cells. And he took a very thin piece of cork from a cork in a wine bottle, and he put that under a microscope, and he saw something like this. Now, what Hooke is observing here are not living cells. This is just from a cork. They're dead cells, but what's left is the cell wall. So what you're seeing here is the outer layer of the cell called the cell wall, and he named these cells. They reminded him of monks' quarters or prisoners' quarters, cells. Around the same time, a Dutch microbiologist named Anton van Leeuwenhoek is the first person to observe living cells. He used a microscope of his own design, this microscope, and that is to scale, to observe a drop of pond water that had been sitting out for a while, and he saw all kinds of little microscopic critters moving around in there. Of course, we know today he's looking at mainly protozoans and algae. However, interestingly, he also saw bacteria. This is Leeuwenhoek's drawings of the different shapes of bacteria we can see here. That's pretty impressive because that means his microscope had to magnify about 1,000 times to see bacteria like that. Of course, now a cell can just, take a selfie, right? So say he's taking a selfie, but he needs a microscope. Anyway, okay, now you have access to these PowerPoints, everyone. So To save time, I'm not going to play you some of the videos I have here in these lecture PowerPoints. But when you review these PowerPoints and you download them to study from and to review for a quiz or an exam, be sure that you click on the video here and watch this. This video here is about Leeuwenhoek and how he discovered cells. And it's important that you watch that. I may ask some questions about it. Okay? Now, if we fast forward past Robert Hooke and Leeuwenhoek, we get to what's called the golden age of microbiology, right here, 1857 to about 1907. Now, why this is called a golden age is this is a period of many new important discoveries that were made in microbiology. So if we look at the time scale here, the yellow period here is about the golden age of microbiology, where a lot was happening. And we'll be looking at other key figures like Louis Pasteur here, Robert Koch here, a little bit later in the course. All right, so one of the things that came out of the golden age of microbiology is answers to age-old questions like, does microbial life spontaneously generate? Okay. Now, spontaneous generation was an ancient belief and actually lasted all the way up to about the 1860s and a little beyond. So for most of human history, people had a belief in spontaneous generation. Spontaneous generation is the belief that life can come from non-living matter. Like life just spontaneously arises or generates from non-living things. Biogenesis is more of the modern concept where life only comes from other life. So for most of human history, dating back to Aristotle, the ancient Greek philosopher way back, for most of human history, spontaneous generation was what people thought. Now, not for more advanced organisms like humans and some major mammals and animals, but for smaller things like worms and insects and frogs and things like that, the belief was they could spontaneously generate. By the late 1600s, though, and especially into the Golden Age, scientists begin to challenge this idea of spontaneous generation. Now, one of the people who did in the 1600s was a French scientist named Francesco Redi, who did an experiment like this. You take raw meat, put it in a glass jar, unsealed, another chunk sealed with a cork, another chunk, and this one no air could get in. Another one here, we put just some cheesecloth over it so air can get in. And only the one that was totally left open did maggots grow. So Redi was able to show that even if you let air go through, okay, we don't get spontaneous generation unless flies can land on the meat and, of course, lay their eggs. And then when we get even further, now Redi's experiment was pretty good, but spontaneous generation still lingered, all right? So by the 1860s, a famous French scientist named Louis Pasteur performed what we now call his swan-necked flask experiment, a very famous experiment that would disprove spontaneous generation. Now, here's what Pasteur did. He took some gravy here that had microbes in it, and he boiled it for a long time, really, really hot. And see this glass tube? It's kind of got this long swan-necked or swan-like bend to it, the neck of a swan. And he boiled it and he killed all microbes. Now, the end here is open to air, so air can get in and out. And he lets it sit and cool down. He checks it. No microbes, no microbes, no microbes, no microbes ever get in here. He finds dust particles down here that do have microbes in them. So what's the significance of the dust settling in the bend of the flask? We have some options here. So which one do we think it is? So the dust traps the microbes, the dust is the source. We're really looking here at number 2. We know the dust is where the microbes came, so microbes did not spontaneously generate from this broth. They had to be brought in from the outside. And this would pretty much definitively prove spontaneous generation. Now, since this time, we have developed cell theory. Cell theory is one of the foundational bedrock theories in modern biology. Cell theory says that all organisms are composed of one or more cells. A cell is the basic organizational unit of life, meaning a cell is the smallest thing that is itself alive, as I told you earlier. And all cells arise from pre-existing cells. Okay? So we've advanced quite a ways here and now have the idea of cells as a foundational concept in biology. All right, now next I want to talk a little bit about the chemistry of microbiology. You might have heard the word matter. Matter is anything that has mass and takes up space, and matter is composed of chemical elements, which we call atoms. So like every atom on the periodic table is a chemical element. 92 of these occur naturally in nature. Many others have been produced by us humans. Now, of all those elements on the periodic table, which ones are most important for living things? You want to remember this acronym, SPONGE. Sulfur, phosphorus, oxygen, nitrogen, carbon, and hydrogen. So these might be the healthiest snacks in the world. See, SPONGE. All right, anyway, close second to these top 6, these SPONGE elements, would be sodium, potassium, calcium, magnesium, chlorine, iron, and iodine. Okay? Atoms, in turn, are composed of smaller particles called subatomic particles. You may have heard of these. Protons are a small particle that makes up part of an atom. They have a positive charge. Neutrons have no charge. And the most important one is electrons have a negative charge. So here we see a picture of a sulfur atom. The blue spheres here are electrons, and the protons and neutrons would be in the center here. Now, electrons are the most important because they are the subatomic particles. They're the part of an atom that interacts to connect atoms together to form what we call molecules or chemical bonds. So 2 or more atoms bonded together, we make a chemical bond that holds them together, and we now call that a molecule. It's the electrons that do all that work. Organic molecules contain carbon. Inorganic molecules do not. So you'll want to know that distinction. And then when we talk about a chemical reaction, we can build something up. This is a synthesis reaction, so like A plus B makes AB bonded together. This is our product, AB. Or we can break something down in a decomposition reaction where we have our AB and we break it into its parts. The pH scale is a way of measuring how acidic or how basic a solution is. So the scale ranges from 0 to 14, with 7 right in the middle being a neutral solution. Below 7 is an acid, and the lower the number, the stronger the acid. And above 7 is a base. The higher the number, the stronger the base. So in this diagram here, you can see some common substances and what their pH might be. A buffer is any substance used to keep pH constant, prevent it from changing too much, or at least beyond a range that we want. Okay, now with that little bit of chemistry in mind, the next key concept are the main macromolecules that living organisms rely on. All right, now macro means big, like micro means small, macro means big. So a macromolecule is a large molecule, and the parts that make them up are called monomers. So I like to use an analogy, like a macromolecule is like a finished Lego product, and each individual Lego is a monomer. Now, there are 4 families of macromolecules that all living things use and depend on. Number 1 are carbohydrates. They are composed of simple sugars. You want to know for each family what the monomers are and what their functions are. So if we hook together a whole bunch of simple sugars, we're going to get a carbohydrate of some kind. There are 3 basic categories. Mono means one, monosaccharide, one sugar. So a monosaccharide is simply one sugar monomer. The most important for living things is glucose, pictured here. Notice glucose is not a straight line, it's not linear, it's bent into a ring. C6H12O6, so 6 carbons, 12 hydrogens, 6 oxygens. Glucose is the easiest source of energy, and we'll be coming back to that later in the course. Disaccharides are the next category of carbohydrates. They are 2 sugar monomers hooked together. Important ones you may be familiar with are lactose. This is found in dairy products, particularly milk. And sucrose is regular table sugar that you might cook with or put on your cereal. And then poly means many. So polysaccharides are 3 or more sugar monomers hooked together. And everybody, it's usually a lot more, like hundreds or thousands of them. Most things that you think of as a carbohydrate, like pasta, bread, rice, potatoes, bakery, donuts, things like that, are polysaccharides. So starch, all your starches are in this category. Cellulose is what makes up the outer layer, the cell wall of plant cells. And chitin makes up the outer layer of fungal cells, like a mold, a yeast, or mushrooms. Now, the functions of carbohydrates include an immediate supply of energy. They're the easiest thing to break down to get energy, and also energy storage, especially your polysaccharides. The next family of macromolecules is lipids. All lipids are insoluble in water. They cannot dissolve in water. And what makes up a lipid, the monomers that make them up, are fatty acids and glycerol. So you take a bunch of fatty acids and a bunch of glycerol and we hook it together, I'm going to get a lipid of some kind. Now, there's also 3 categories of lipids. What you might be most familiar with is the first category, which is fats and oils, and some products pictured here. Fats and oils are composed of triglycerides, which is 1 glycerol, and so you have 1 glycerol and then 3 fatty acids hooked to it. Okay, so I've got 3 fatty acids hooked to that 1 glycerol, and I put a whole bunch of triglycerides together and I get a fat or an oil. Functions include a reserve supply of energy. After carbs, they're the next most easy to break down. Heat or thermal insulation, and shock absorption. Phospholipids, our next category, are composed of diglycerides. Now we have our 1 glycerol, 2 fatty acids, and in place of that 3rd fatty acid, we have a phosphorus. Their function is to form membranes. The membrane around every cell, not the cell wall, but the membrane just inside that, is made up mostly of phospholipids. And lastly, sterols. Sterols are lipids that are not a straight line. They're bent into a ring structure like this. So cholesterol, note that the word cholesterol has sterol in it. Okay, you can see the 4 rings. So cholesterol is an example. The sex hormones, testosterone, estrogen, progesterone, are all sterols, as are waxes, earwax, beeswax, candle wax, things like that. Functions here, cholesterol functions in membrane structure. Hormones are chemical messengers. They deliver a message from one part of the body to another. And protection and waterproofing is going to be your waxes. Proteins. Our third family of macromolecules are proteins. Some of the things pictured here you might recognize as what you think of as proteins. Now, what makes up a protein, the individual parts that make up a protein, the monomers, are called amino acids. So here's an amino acid. You can see he's being really mean here. Or this guy. What do you call an acid with an attitude? Give me your lunch. Amino acid. All right, okay. But anyway, there are 20 different major amino acids, 2 others that are less common. And amino acids are hooked together using what's called a peptide bond, which is carbon to nitrogen. The biggest function of proteins is that they are the building blocks of life. They provide structure. They're the structural basis of life. But proteins have a lot of other functions. They include enzymes. Enzymes are proteins that make reactions occur faster. We call them catalysts. A catalyst is something that makes a reaction occur faster. And enzymes are a type of catalyst. Many other functions. Many hormones are proteins. Hemoglobin, which is a molecule on your red blood cells that transports oxygen throughout the body, is a protein. Antibodies, little proteins that fend off disease. And collagen, a connective tissue that literally is kind of the glue that holds the body together, is a protein. Proteins can have different levels of structure called primary, secondary, tertiary, even quaternary. Now, I'm not going to go into that or hold you responsible for that, but you want to know that proteins can take on different levels of structure. Our last family of macromolecules is nucleic acids. Now, the monomers are nucleotides. Everything in this white box is one nucleotide. It has a nitrogen base up here, a sugar here, and a phosphorus here. Now, if I hook together a whole bunch of these, I'm going to get a nucleic acid of some kind. There are 3. RNA, ribonucleic acid. Look how we've hooked together 4 nucleotides here. DNA, which is a little bit bigger and thicker than RNA, deoxyribonucleic acid, and ATP. ATP represents chemical energy in living organisms. So we'll be coming back to all of these later. What you know of as a gene is a functional piece of DNA. It's a piece of DNA that does something. Overall, functions of DNA and RNA include inheritance, it's passed on to offspring, and they determine the structure of proteins, something we'll get to in the chapter on microbial genetics. I already mentioned that ATP is chemical energy in living organisms. And then lastly, everyone, by way of a little bit of finishing final chemistry here, cells and solutions. A solvent is a liquid that dissolves another substance, whereas the solute is the substance that gets dissolved. So if I take a teaspoon of sugar and mix it in a glass of water, water's the solvent and the sugar's the solute. The final product is the solution, the solute plus the solvent. Okay? Hopefully that makes some sense and gives you a little overview here of microbiology. The next chapter will go into a little bit more on tools that we use in the laboratory and how we categorize life on Earth. So until next time, everyone, do remember you have access to handouts for each chapter and the PowerPoints here that I was just going over. Okay, until next time, have a good rest of the day, everyone.